and today I will give a talk about how to maximize the performance of DNA analysis algorithms using aspects of the Apache Arrow project. DNA analysis has shown to have immense societal value with wide-ranging applications from healthcare, agriculture, livestock improvements, and much more. It's hard to find aspects of our daily lives today that are not touched by the genomics revolution. Various applications like the recently approved, FDA-approved gene therapy for human disease to the tracking of the spread of the coronavirus using its DNA sequence and accelerating development of vaccine to it using this data. However, with the continued increase in the amount of data and the exponential increase in the available data sets, a lot of challenges continue to arise. More and more complex compute methods are used that have compute times ranging from hours to days, and the large amounts of data from terabytes to petabytes that needs to be stored and processed. This means that there are a lot of opportunities to reduce the computational cost and improve computational efficiency of our DNA analysis pipelines. One of the most widely used DNA analysis workflows is called the GATK workflow, which is the Genome Analysis Toolkit developed by the Broad Institute, used widely for cancer DNA analysis, for example, to identify mutations or variants in the DNA sample of a patient. GATK uses a number of traditional computational techniques like storing data in disk and repeatedly accessing it from disk, analyzing data in tabular row-oriented formats, and also using multiple programming languages to implement various different stages in the workflow and having these different languages accessing the same data. These different aspects or limitations represent hallmarks of the improvements that the Apache Arrow project can provide to these kinds of big data analysis workflows. To start, we propose a new file format that is based on the Apache Arrow project that replaces standard existing file formats in the field, in the genomics field. One of the most widely used file formats is the SAM file format, the sequence alignment map format, which is a standard tabular DNA sequence file that is row-oriented and is text-based. We replace it with a columnar representation we call the ArrowSAM format that has a schema to allow you to access specific fields in the file without having to access the whole file in order to read the data included within it. And then we use this ArrowSAM format to modify the GATK workflow so that it benefits from the available characteristics of the Apache Arrow project. A standard GATK workflow starts with patient DNA and a reference DNA to compare the patient DNA with and identify mutations or variants. And then this data is stored in disk and then accessed multiple times by various different stages of the GATK workflow. We start by replacing the SAM file with the ArrowSAM file in memory. This allows us to reduce the accesses to a disk significantly by keeping the data in memory the whole time as the pipeline is processing the data until we reach the end of the pipeline and identify the variants. In addition, we use the Apache Arrow APIs to be able to access this data in the memory without having to modify it or copy it, which allows us to access the data in different programming languages such as Java, C, and Python that are used to implement the various different stages of the GATK workflow. To give an example of how the GATK pipeline increases in efficiency as we use the columnar data format, this figure shows how many fields in the ArrowSAM file are being modified by the different stages of the GATK workflow. So we see that in the first stage, all the different fields are instantiated or created. And then in the second stage, all these different fields are modified. But then in subsequent stages, in the 4 subsequent stages, these fields are barely touched. Most of the time, almost all the fields are either only red, represented by the gray color, or never even accessed, represented by the red colored blocks. Which means that storing the data in this columnar format is very beneficial to optimizing how many times we access the data in the memory for DNA analysis pipelines, such as GATK. Comparing the resulting pipelines with the baseline pipeline, we see that, for example, on the left-hand side, the CPU utilization of the two different pipelines, the one GATK with Apache Arrow and below the one GATK without Apache Arrow, we see that CPU utilization in the sorting and mark duplication stages, which are difficult stages with low utilization usually in the baseline workflow, have much higher CPU utilization when we combine it with Apache Arrow, which means that we are using our computational infrastructure much more efficiently to be able to perform our analysis. If we compare the runtime of our Arrow-based pipeline with the standard GATK pipeline, we see that our pipeline has the shortest runtime compared to any variant or any type of GATK pipeline, even if we optimize it significantly by storing all the data into memory using the RAM disk approach, by just dumping the data into memory from disk. Even if we compare our GATK Arrow-based file pipeline with custom-made pipelines such as LPrep, which uses in-memory data access, we see that we are still a little bit faster than the fastest optimized version of it, but with a large improvement in the data storage, in the data memory utilization efficiency. We use 4 times less memory compared to the memory requirements of LPrep. Comparing the way we access the data in the memory, we see that the ArrowSAM format increases memory access efficiency significantly. We see that we can utilize much more efficiently the whole memory hierarchy in our compute system, all the way from accessing main memory or accessing the various different levels of cache, like level 1 and level 2 in the processor. This figure shows that our ArrowSAM based workflow is able to reduce memory accesses by more than an order of magnitude, thereby focusing the effort on processing the data in the processor rather than waiting for the data to be accessed in memory. In conclusion, we presented a new file format for DNA analysis called the ArrowSAM format based on the Apache Arrow project that significantly improves runtime and increases efficiency of DNA analysis. We showed that we are able to use the various aspects of the Apache Arrow project to address many limitations of DNA analysis pipelines, such as in-disk storage, tabular row-oriented data representations, and using multiple programming languages to access the same data in the memory. We also showed that the ArrowSAM format allows to increase the speed of the difficult stages of the workflow by more than 5 times, and is able to increase the speed of the full pipeline by up to 1.5 times. And finally, the ArrowSAM format allows us to have 4 times more memory efficiency, access efficiency, than even optimized custom-made in-memory workflows that can process the same data. Thank you very much for your attention, and goodbye.